Welcome to the Deep Dive, your shortcut to being exceptionally well-informed on the bleeding edge of science and AI. Today, we're plunging into a truly monumental piece of work, a comprehensive paper from Archie Garve. It meticulously maps the rapidly evolving landscape of scientific large language models, or SciLLMs, and multimodal large language models, SciMLLMs. Think of it as a definitive guide to how AI is fundamentally reshaping scientific discovery. We're talking about insights drawn from rigorous research, places like the Shanghai Artificial Intelligence Laboratory, and revolutionary initiatives such as intern discovery, agentic science. Our mission today is to cut through this huge volume of information, delivering only the most critical insights from this extensive review, tailor-made for you.
That's right. And this deep dive is crafted specifically for those of you already well-versed in machine learning, data science, and mathematics. The paper we're exploring provides a professional and comprehensive overview. It deliberately focuses on the established concepts, ideas, and results directly from the source material. We're not venturing into speculation here. We'll introduce the main findings, then dive deep into the intricate mechanisms, and conclude with a succinct summary. It's a highly structured journey through the paper's profound insights.
And when we say comprehensive, we really mean it. This isn't the work of just one or two researchers. It's a collaborative effort by Zhuang and a multitude of co-authors, which really underscores its authoritative nature. It's a fascinating blend of how AI is evolving, the inherent challenges of wrangling scientific data, and the, frankly, astonishing array of domain-specific applications these models are enabling. Okay, let's unpack this and dive into what feels like a new era of scientific exploration. So let's kick things off with what feels like a fundamental revolution. The paper really emphasizes that large language models are no longer just tools for, generating text or having a chat. They are rapidly evolving into sophisticated scientific agents, capable of not just assisting, but actually leading scientific discovery. This strikes me as a monumental departure from our traditional reliance on symbolic modeling and numerical simulation. What exactly defines these scientific agents, according to the paper?
It's a critical distinction, and yes, the paper truly highlights this paradigm shift. What's fascinating is how it outlines the characteristics defining these agents. We've seen this incredibly rapid evolution, right, from models like BERT back in 2019, which were primarily focused on understanding text, to advanced systems projected for 2025, like GPT-5 perhaps, demonstrating a profound increase in scientific capability. And this isn't just about language anymore, it's about a progression through knowledge integration, task adaptation, and ultimately what the paper terms agentic science. These SciLLMs and SciMLLMs are becoming capable of processing natural language descriptions, really complex physical systems. They can accurately estimate physical parameters, simulate dynamic evolution, and even represent physical laws symbolically.
Wow. Symbolically, even.
Yes, and this capability has fostered the emergence of what they call AI co-scientist roles. We see powerful examples like Gemini 2.0 and Co-Scientist, which uses GPT-4, being highlighted, and also promising initiatives like Intern Agent. It really is a leap from tools to collaborators.
That's a huge jump in capability. Moving beyond just a single powerful model, it sounds like these aren't just isolated geniuses, but maybe entire research teams in miniature. Can you elaborate on how the paper describes this concept of multi-agent collaboration and how these agents actually do science?
Absolutely. The paper does discuss how multiple AI agents can indeed work together, precisely mirroring the collaborative dynamics you see in human research teams. And they do this to effectively tackle highly complex scientific problems. It goes beyond just one model's reasoning. A critical aspect here is tool use and orchestration. This is key. The paper cites significant advances in integrating LLMs with external scientific tools, databases, instruments, the works. This includes things like knowledge graph-driven orchestration and developing domain-scale agents. Imagine agents interfacing with hundreds of software tools, databases, lab instruments, all while meticulously tracking provenance.
Provenance meaning where everything came from.
Exactly, where every piece of data, every step originated. This allows agents to transcend their internal knowledge stores and actively interact with real-world scientific workflows. Models like ChemCrow, based on GPT-4, exemplify this. It enables end-to-end tasking from, say, retrosynthesis, basically reverse engineering a chemical synthesis, all the way to catalyst design. And Co-Scientist, also GPT-4 powered, pushes this even further. It combines advanced planning with actual code execution and instrument control in cloud laboratories. Think autonomous chemistry experiments. It's about moving from purely digital tasks to tangible interaction with the physical world.
The idea of models interacting with actual lab instruments, that's genuinely impressive. But for something to be a true agent, it needs to be able to learn, right? To improve. Does the paper address how these systems evolve over time? Maybe even teach themselves?
That's a very important point. And yes, the paper delves into the concept of self-evolving agents. These are systems designed to continually adapt and improve their performance over time. It describes these continual adaptation loops within the typical plan, experiment, and verify workflow that's inherent to scientific research. So what this means is that agents externalize feedback from their experiments. They update their episodic memory, essentially, what they've learned from past experiences. They progressively accumulate executable skills, much like a human scientist refines their techniques over years. An interesting example given is Voyager, which builds a growing library of programs and an automatic curriculum that can even be transferred to new environments.
So it learns reusable skills.
Precisely. And these agents can even create their own tools. The paper mentions Toolformer, which self-supervises the acquisition of API calling skills that persist across various tasks. These mechanisms allow agents to learn autonomously from experience, expand their capabilities, become more robust over long operational horizons. They effectively become better scientists over time.
So it's not just executing tasks, but constantly refining those tasks, maybe even inventing new ways to achieve scientific goals. Given all this, what's the ultimate aspiration for these scientific agents, according to the paper? What's the grand vision here?
The ultimate goal, as the paper outlines it, is autonomous scientific discovery. This envisions a future where these agents can independently design, execute, and analyze experiments, ultimately leading to novel scientific breakthroughs without constant human intervention in every single step. This is really the pinnacle of their evolutionary path, moving towards a truly agentic science where AI systems contribute directly and autonomously to advancing human knowledge. And the velocity of this development, going from basic text understanding to this level of agentic science in just a few years, it really underscores the transformative potential.
So we've painted this picture of incredible scientific AI agents, but every brilliant mind needs fuel, right? And for AI, that fuel is data. What struck me in this paper is how fundamentally different scientific data is from the kind of text general LLMs feast on. The paper identifies a critical data bottleneck as a driving force behind the rise of scientific agents. Can you walk us through what makes scientific data such a unique beast?
You've absolutely hit on the core challenge and the opportunity. The distinction is paramount. Because scientific data exhibits extreme heterogeneity across modalities and formats, it's far more complex than the relatively homogeneous text corpora used for general-purpose LLMs. What truly separates scientific AI's data bedrock is its, well, almost fanatical devotion to provenance and micro-level detail. That's a stark contrast to the vast, often uncurated web data general LLMs use.
Okay, so heterogeneity in detail. Let's break down these diverse data types. Where do we start?
We start with textual formats. These really form the foundational substrate. This includes primary experimental documentation, think laboratory notebooks, experimental protocols, field observations. These raw records are invaluable because they capture the entire discovery process, including the failures, which are crucial for reproducibility and understanding scientific reasoning. This primary data then feeds into highly specialized databases central to modern science. We're talking petabytes of structured information in places like GenBank for genomic sequences, RCSB for protein structures, PubChem for chemical compounds, NASA's ADS for astronomical observations.
Huge structured repositories.
Exactly, and beyond these, domain-specific training corpora are essential, like Cybert, pre-trained on millions of full-text scientific papers, including Physics Lit, or AstroBert, leveraging the NASA ADS for astronomy literature. The depth and specialization here are immense.
That's a massive amount of highly specialized text, but as you mentioned, science is far more than just text. What about the myriad of visual modalities these models are learning from?
Visual data. It's incredibly diverse and information-rich in scientific contexts. First, consider spectroscopy. Data from UVVs, infrared, Raman, NMR, they serve as molecular fingerprints across chemistry, material science, physics, and the visual representations of these spectra have proven highly effective for AI-driven spectrum learning. Then in life sciences, we encounter various forms of microscopy. Light microscopy captures cellular structures, fluorescence microscopy details protein localizations. Datasets like the Human Protein Atlas support cell segmentation, phenotype classification. These images, often in specialized formats like TIFF or ND2, are key for visual language models. And then you have whole slide digital pathology images. These can be gigapixels in size, essential for cancer diagnosis. Large cohorts provide thousands of images paired with diagnostic reports.
And beyond the microscopic, medical applications seem like a huge area.
Absolutely. In medical imaging and healthcare, the visual modalities are extensive and crucial. This includes ophthalmology, with fundus photography and OCT for eye conditions, dermatology for skin lesion analysis, ophthalmic surgical microscopy for high-res intraoperative visualization, endoscopy for surgical guidance. When these visual data are paired with descriptions and reports, they hold immense potential for developing powerful healthcare MLLMs. On a much larger scale, you have macroscopic and remote sensing data, natural photographs for biodiversity, agricultural visual data from UAVs and satellites for crop monitoring, and earth science leveraging satellite remote sensing and atmospheric datasets for climate modeling. What's critical here, especially for Earth observation, is the diversity of collection sources. You have ground-based stations, multispectral remote sensing, reanalysis data integrating observations with models, each offering unique features in spatial coverage, temporal resolution, and observational content.
That gives us a truly global and even microscopic perspective. Thinking even bigger, what about astronomical visual data? We all see those incredible images from NASA and ESA.
Indeed. The paper highlights stunning examples. It showcases the application of radio, optical infrared signals in imaging various celestial objects. Visuals like the W50 Manatee Nebula, the galaxy cluster ACOS 295, the NGC 4535 spiral galaxy, all imaged by instruments like the VLA, Hubble, James Webb Space Telescope. These visuals are critical not just for us humans, but for AI to learn to classify, analyze, and maybe even discover new cosmic phenomena.
Fascinating. So we have textual, visual, but science also heavily relies on abstract representations. What does the paper tell us about symbolic representations?
Symbolic representations are a fundamental data modality. They offer abstract, non-numeric encodings of scientific entities, relationships, laws. They're designed to be both human interpretable and machine processable. The core idea is to preserve semantic meaning for automated reasoning. In chemistry, this includes molecular structures encoded as string notations like smiles or inch-I, and even things like selfies, specifically designed to be robust for chemical modifications. For material science, crystallographic information files, or CIFs, are the standard. They encode unit cell parameters, atomic positions, symmetries, experimental metadata. These underpin major databases. ZINC, Chimbiel, USPTO, the Materials Project. In biology, symbolic representations extend to DNA, RNA, protein sequences, the building blocks of life. These abstract forms allow AI to manipulate and reason about scientific concepts in a structured, logical way.
The granularity of that information is incredible. Now, much of science is about understanding change over time. What about time series data?
Time series data is crucial for capturing dynamics across multiple scales. The paper emphasizes its multi-scale nature. In biological systems, this data captures dynamics from molecular-level gene expression patterns over time, all the way to clinical monitoring through ECG, EMG for muscle activity, continuous glucose monitoring in diabetic patients. Neuroimaging modalities provide complementary temporal and spatial resolutions. You have fMRI detecting bold signals, mapping brain networks, and MEG measuring magnetic fields from neuronal activity with millisecond precision. In chemistry, molecular spectrum data Raman, IR, UV, NMR reveal structural info as a function of wavelength or frequency, enabling AI-driven analysis. Physics leverages high-frequency data, like strain data from LIGO Virgo at thousands of hertz for gravitational wave detection. For Earth science, spatiotemporal context is particularly critical. Global meteorological variables might be represented as a large tensor for spatial discretization, while temporal resolution varies massively. Nowcasting might use 10-minute intervals, medium-range forecasting maybe every 6 hours.
It sounds like an orchestra of data, all playing different, incredibly detailed parts. And in the life sciences, we often hear about multi-omics data. How does the paper characterize this, and why is it so important?
Right, the paper describes the multi-omics data landscape in life sciences. This covers genomics, transcriptomics, proteomics, metabolomics, the microbiome, even the exposome. These different omics layers are interconnected through biological processes, DNA to RNA to proteins. They offer complementary insights. When combined, they provide a far more comprehensive understanding of biological systems than any single layer alone.
So it's about the whole picture.
It's exactly. Focusing on transcriptomics, which captures gene expression, its data ecosystem includes transcript annotations, repositories for RNA-seq profiles like GEO, expression atlases like the Human Cell Atlas. Crucially, spatial transcriptomics platforms like 10x Genomics Visium link gene expression profiles to precise tissue coordinates. This enables spatially resolved analyses that were impossible before. For proteomics, the field juggles molecular representations from distinct channels. Structured knowledge bases like UniProt KB for sequences, the Protein Data Bank, PDB for 3D folds, plus interaction networks like String and BioGrid, which fuse biochemical and genetic evidence to build genome-wide association graphs revealing protein interactions. This integration across omics layers is vital for AI to truly understand complex biological systems.
That's an astonishing level of detail across so many different data types. Given this incredible diversity, how does the paper address the unique challenges of integrating all this data, especially for specialized areas like remote sensing?
The challenge of data integration is indeed significant, particularly because the unique characteristics of, say, remote sensing RS imagery necessitate specialized datasets and sophisticated fusion techniques. The paper points to initiatives like Eagle Vision, trained on EVAT-Hull 95K, the first large-scale dataset specifically for fine-grained object-level understanding in RS imagery. This allows for comprehending intricate object attributes beyond just simple classification. Similarly, EarthMarker is supported by the RSVP dataset containing roughly 3.65 million multimodal pairs of image point text and image region text. This enables nuanced interpretations guided by visual prompts. These examples highlight the ongoing effort to develop highly specialized datasets and advanced integration techniques. It's about handling the unique properties and semantic complexities of different scientific modalities. It's not just about collecting data, it's about making it all speak the same language, essentially. #3. Key challenges and quality standards in scientific AI.
Okay, so we've explored the incredible capabilities these scientific AI agents are developing and the vast heterogeneous data ecosystems they rely on. But what does this all mean for building truly robust scientific AI? The paper highlights critical challenges stemming directly from the complex nature of scientific reasoning and the data itself.
Precisely. The paper identifies three fundamental challenges. The first, and arguably one of the most significant, is interpretability in scientific AI. The inherent black box nature of many LLMs and MLLMs is a major bottleneck. Because scientific reasoning inherently demands clear explanations and justifications, the why and how behind a conclusion. This is absolutely crucial for building trust, especially in high-stakes fields like drug discovery, where understanding the model's rationale is just as important as the output itself.
You need to know why it thinks that drug works.
Exactly. The paper discusses chain of thought, or COCI, as an emerging technique to elicit explicit natural language reasoning traces from models. BioReason, for instance, applies this multi-step reasoning to DNA foundation models, integrating them with LLMs for deep interpretable biological reasoning from complex genomic data. However, conducting COCI reasoning in scientific AI models is particularly challenging. It's due to the inherent complexity and domain-specific nature of scientific knowledge spanning biology, chemistry, physics. It requires hypothesis-driven logic grounded in empirical evidence. It's a completely different level of why than in general AI.
So getting the why is harder and far more critical than just getting the what. What are the other major challenges the paper pinpoints?
The second challenge is multimodal integration. We touched on this, the inherent difficulty of seamlessly combining all those diverse data types. This goes beyond simply concatenating data. It requires deep semantic alignment, understanding how different modalities relate scientifically. And third, there's dynamic knowledge evolvement. How do you ensure these models can continually update themselves with the rapidly changing landscape of scientific discoveries? Without constant, expensive retraining from scratch, that is. Scientific knowledge isn't static. It's a living entity, and models must reflect this dynamism to stay relevant.
Those are indeed significant hurdles. But even before we get to those challenges, we have to consider the input itself. What about the quality of the data feeding these powerful models? The paper outlines specific, rigorous quality standards for scientific data sets. How do we ensure the data these agents are learning from is actually good?
An excellent question. The paper outlines 4 complementary dimensions for data quality, all essential for robust scientific AI. The first is accuracy. This assesses how faithfully the data represents the underlying phenomena, whether it's spatial positioning and remote sensing, temporal annotation in time series, or signal fidelity in spectroscopy. High accuracy boosts training efficiency, inference precision, and directly impacts the credibility of scientific conclusions. For example, Landsat 8 satellite imagery, after correction, achieves geolocation errors of just 15 to 30 meters. That's remarkable spatial precision. In physical sciences, the Materials Project provides data from first principles calculations, meticulously controlling errors for properties like band structure. Accuracy is typically quantified using metrics like MSE, RMSE, signal-to-noise ratio against reliable ground truth, or benchmarks. It's about being precise enough for science.
That makes perfect sense. You can't build good science on shaky data. What about making sure the data set covers everything it needs to?
That brings us to the second dimension, completeness. This refers to the extent a dataset captures all relevant elements across content, structural fields, temporal coverage. In genomics, for instance, completeness is evaluated by sequencing depth. Modern standards require average coverage of 30x or more. Below 10x is often insufficient to accurately detect mutations. In materials science, Data integrity, its completeness, directly determines the success of data-driven discovery. Methods for assessing this include missing value statistics, field coverage analysis, time series gap identification. Earth science datasets like SCDNA show how crucial filling in missing meteorological data is for integrity. Tools like OpenRefine can help automatically detect missing entries. It's about striving for the full picture.
And given the rapid pace of scientific discovery, how important is timeliness for these data threats?
Timeliness is absolutely critical, especially for dynamic applications like monitoring or forecasting. This dimension measures data update frequency, the latency between collection and release, and how quickly data responds to real-world changes. It's crucial for things like emergency response, real-time health monitoring, trend forecasting. For example, during the COVID-19 pandemic, the Johns Hopkins dataset was updated daily, enabling rapid modeling and public health responses. NASA's MODIS satellite products are updated daily for environmental monitoring. This contrasts sharply with more static datasets like ImageNet, which, while foundational, are designed for real-time relevance.
Finally, for reproducibility and trust, we need to know where the data comes from and how it's been handled. What's the fourth quality standard?
The fourth dimension is traceability. This involves meticulously documenting the provenance, the origin, and history, plus metadata, version histories for transparency and reproducibility. Traceability is absolutely crucial for evaluating reliability, usability, long-term value. Without proper provenance, it becomes incredibly difficult to verify the data's origin, the experimental conditions, or the processing steps. This fundamentally undermines trust and reproducibility in scientific AI, which are non-negotiable in scientific discovery. These four dimensions, accuracy, completeness, timeliness, traceability, collectively standardize data management and guide optimal AI deployment. They ensure the bedrock is solid.
We've established the ambition of scientific AI agents and the rigorous demands on their data, but how do we actually measure their performance? Evaluating these scientific models seems vastly different from evaluating general-purpose LLMs. It feels like the stakes and the requirements are fundamentally higher. What makes evaluating scientific AI models a whole new ballgame?
You're right, the distinction in evaluation frameworks is fundamental. It's not the same game at all. General LLM benchmarks primarily assess core NLP abilities and general reasoning. Things like language understanding, fluency, factual recall, generic problem solving. They're designed for broad linguistic competence, general cognitive skills, often assuming only basic computational skills and maybe high school level science. Factuality is usually grounded in general web content.
Okay, standard stuff.
Exactly. In stark contrast, scientific LLM benchmarks demand mastery of depth, precision, and rigor, the hallmarks of academic research. They go far beyond general linguistic competence. These benchmarks evaluate deep, domain-specific scientific knowledge, the ability to reason with formal systems equations, symbolic logic, the capacity to retrieve and synthesize scholarly information from complex literature. And critically, their support for hypothesis generation or experimental design. It's about how they interact with and advance the scientific process, not just how well they communicate.
That makes perfect sense. The bar is simply much higher. Can you give us some examples of what this expert-level scientific knowledge comprehension and retrieval actually entails? It sounds like it goes well beyond answering simple trivia questions.
Right, it's a critical dimension because it requires models to retrieve, comprehend, and apply cutting-edge research knowledge across diverse scientific disciplines with true domain-level expertise. This extends beyond general facts to include domain-specific equations, physical constants, technical terminology, theoretical constructs, you name it. The paper highlights this as a cornerstone for enabling automated scientific discovery. Key evaluation aspects include sophisticated information retrieval, rigorous literature-based fact verification, and seamlessly integrating heterogeneous scientific knowledge. For instance, SciVench introduces tasks requiring retrieval and application of complex mathematical equations, chemical laws, physical theorems. SciNoVol spans domains from biology to materials science, assessing tasks like precise molecule identification and multi-step reaction prediction. And SciQA leverages the Open Research Knowledge Graph to support complex cross-domain questions requiring synthesis of information from disparate fields. This dimension truly challenges models on both breadth and depth, emphasizing accuracy, completeness, and engaging with knowledge beyond the surface level.
Okay, this is where the rubble meets the road for our expert listeners. What about the advanced evaluation metrics and methodologies themselves, beyond just a simple accuracy score? When you're dealing with such complex data, simple accuracy probably doesn't cut it.
Indeed, the paper delves into this extensively. We're seeing a clear evolution beyond simple accuracy towards highly domain-specific measures. For biological data, metrics like AUROC and MCC are standard for imbalanced classification tasks, common in genomics. For regression tasks in genomics and multiomics, maybe in data sets like diseases or RepoBB, R-squared, RMSE, and Pearson's correlation coefficients, PCC, are widely used to assess linear relationships. In chemistry, evaluation for molecular generation goes beyond mere structure. It emphasizes chemical validity, drug-likeness, basically assessing if newly generated molecules are synthesizable and pharmacologically plausible, often judged by human experts.
So could a chemist actually make this molecule?
Precisely. For physics, metric specialization happens along two axes. One is exact string or structure match. For symbolic regression, does the discovered equation match the ground truth? The other, crucially, is stepwise or explanation-sensitive grading. This penalizes reasoning drift even if the final answer is numerically correct, meaning the path to the answer is as important as the answer itself.
That's a much more nuanced approach.
It has to be. And for multimodal tasks, visual grounding uses metrics like intersection over union, IOU, to assess how well a model localizes objects within images. The merit of this trend is clear. Metrics are increasingly aligned with actual scientific objectives. They're designed to reveal specific failure modes that generic QA accuracy would simply hide.
That's a much more scientifically rigorous approach to evaluation. I'm also curious about this idea of an agent evaluating another agent. The paper mentions an agent as a judge paradigm. Can you explain that? How might it be more effective than traditional benchmarks?
Yes, the agent as a judge paradigm. What's fascinating here is this emerging trend for evaluating these complex agentic systems. With the rapid advancements in LLMs and multimodal models, traditional evaluation, often relying on a single number or lots of manual labor, is proving inadequate. This paradigm extends the LLM as a judge idea by incorporating agentic features like dynamic planning and intermediate feedback. So instead of a fixed one-shot evaluation, it employs a multi-round dynamically adjusting process that sort of mimics human experts. An agent judge can adapt its evaluation direction and test cases based on intermediate results and observed feedback.
So it's more interactive.
Exactly. This reduces reliance on static benchmarks at large, expensive sample sizes. For instance, in code generation, the agent judge evaluates multi-step task performance, not just the final code. For visual generation, an evaluation agent can provide detailed natural language analysis, not just a score. In hypothesis generation, a judge agent can evaluate novelty, validity, coverage of proposed hypotheses, much better suited to their open-ended nature. This framework provides richer, more reliable evaluations by analyzing the entire process and reasoning, not just the final output. It's assessing the scientific process.
So it's not just about the answer, but the scientific journey to get there. What about scenarios where ground truth is hard to come by, which I imagine happens a lot in cutting-edge scientific discovery?
That leads us directly to test time learning, TTL, and its value proposition in natural sciences is significant. Scientific benchmarks often lack definitive ground truth answers, right? This perfectly fits TTL's paradigm of adaptation at inference time without needing labeled data. Furthermore, scientific datasets inherently exhibit strong heterogeneity and often suffer from distribution shifts. Think about earth sciences, atmospheric, oceanic, remote sensing text, huge differences in data structure. Statically pre-trained LLMs often underperform when faced with such shifts. TTL enables immediate adaptation by dynamically updating parameters or reasoning strategies using the currently observed unlabeled test samples.
It learns on the fly from the test data itself.
In a sense, yes, it adapts. Practical applications are diverse. MedAdapter, for example, uses post-hoc adapters for TTL in biomedical tasks, significantly improving both white box and black box LLMs. In chemistry, scaling test time training with reinforcement learning for chemical language models has shown improved chemical space exploration, leading to novel compounds. And in theoretical physics, a symbolic weak verifier framework within TTL enhances performance on complex physics datasets. This really highlights TTL's crucial role in enabling models to generalize effectively and perform robustly, even facing scientific data complexity and uncertainty. #Type5. Domain-specific SciLLMs and their applications.
Here's where it gets really interesting. We've talked about the principles, the data, the evaluation. Now let's look at how these models are being tailored for specific scientific disciplines. The paper shows research scopes across physics, chemistry, material science, life sciences, earth science, astronomy. Let's dive deep into some fascinating examples. Where should we start?
Well, starting with physics, we see a significant shift. LLMs aren't just for language understanding anymore. They're being integrated with physics engines, visual modules. This allows them to process natural language descriptions of complex physical systems, explicitly estimate physical parameters, simulate dynamics, represent physical laws symbolically. The field is rapidly moving towards these models directly interacting with and influencing scientific workflows, not just passively processing info. This means AI can help physicists not just understand, but actively explore and predict physical phenomena.
That's a powerful integration, moving from description to active simulation. What about chemistry? A field so reliant on precise structures and reactions where a single atom out of place changes everything.
In chemistry, the architectures and data are quite diverse. Molecular structures, reaction data, spectroscopic info, vast scientific literature. One fascinating example the paper highlights is Chem 3D LLM. This model tackles the incredible complexity of generating 3D molecular conformations. That's crucial because a molecule's exact 3D shape dictates its function. It introduces a reversible text encoding of 3D structures and uses reinforcement learning with chemical validity rewards. This ensures the generated structures are not only novel, but also physically plausible.
So it designs molecules that could actually exist and work.
That's the goal. It's a potential game changer for structure-based drug design. AI can propose entirely new molecules that fit a target protein, not just tweak existing ones. Other notable models include ChemDFM, pre-trained on billions of chemical tokens for natural dialogue over chemical knowledge, speeding up reasoning for complex reactions. The applications here are extensive: molecular design, reaction prediction, retrosynthesis, catalyst discovery, quantum chemistry, material science.
From the very small to the very large, like material science, how are LLMs being applied there to discover new materials with specific properties?
In material science, LLMs are heavily explored for representation learning and knowledge integration. A key area is representation learning. Models like Polybert use a DiBerta-based encoder trained on 100 million hypothetical polymer SMILE strings. This lets the model learn deep, abstract representations of polymers, crucial for designing new plastics or composites with desired strengths or thermal properties. Another powerful application is knowledge integration. Qwen2-KG, for example, uses a large model combined with a retrieved materials knowledge graph. This approach hits over 91% accuracy on QA benchmarks by combining chain-of-thought reasoning with structured graph facts. It significantly outperforms LLM-only methods. This means AI can effectively synthesize knowledge from decades of research to propose new compounds or predict their behavior, accelerating novel materials discovery.
The interconnectedness across these domains is becoming incredibly clear. Let's delve into life sciences, maybe starting with molecular biology and drug discovery, an area that could literally change lives.
In life sciences, particularly molecular biology and drug discovery, the advancements are truly significant. We have genomic foundation models like EVO, trained on over 80,000 bacterial and archaeal genomes. That's 300 billion nucleotide tokens. EVO establishes scaling laws for DNA, integrates across DNA, RNA, proteins, and achieves impressive zero-shot function prediction for genes it's never seen before. Its successor, EVO2, scales even further to 9.3 trillion DNA bases, extending context to genome scale, predicting impacts of genetic variations across whole organisms.
Trillion, that's immense.
It's staggering. For proteomics foundation models, the landscape is also evolving rapidly. ESM-Fold, for example, enables direct single-sequence atomic-level 3D structure prediction. It transforms our ability to understand protein shapes just from their amino acid sequences. ESM3 takes this further, unifying sequence, structure, and function in a single multimodal generative model. It allows AI to essentially think about proteins holistically. This accelerates drug discovery and optimization, predicting bioactivity, reducing synthesis trials by maybe 50–70%, as seen with models like Temformer designing COVID-19 inhibitors. These models are essentially learning the complex language of life itself.
It's astounding how these models are accelerating the pace of drug discovery. What about the broader healthcare and medical science applications, moving from the molecular to the clinical setting?
In healthcare and medical science, many recent LMs are adapted from general-purpose models, then further pre-trained on vast domain-specific corpora. Clinical reports, medical literature, imaging data. They're then fine-tuned with medical instruction response pairs. Examples include BioMistral, BioMedLM, the MedPolM series, instruction tuned on large models using prompt engineering for medical Q&A. Crucially, scaling up pre-training is vital. PMC Almo pre-trained on nearly 5 million biomedical papers and 30,000 medical textbooks. Huachuan GPT-2 used over 5 million medical documents. Huachuan M1, trained from scratch on 20 trillion tokens including medical data, shows significant performance across many medical benchmarks.
And healthcare is inherently multimodal.
Exactly. Clinical practice integrates patient descriptions, physical signs, imaging, lab findings. This drives the development of multimodal medical LLMs, MLLMs. These mainly focus on medical report generation and medical visual question answering, VQA. LovaMed and Radiology LMA2 are pioneering works transferring general MLLM capabilities to biomedicine, understanding medical images, generating detailed reports. These models are designed to assist clinicians in complex diagnostic and treatment planning scenarios.
Now, for our listeners interested in how these models interact with the human brain, tell us about neuroscience. This sounds like a frontier where AI could unlock some incredible secrets.
In neuroscience, pre-training primarily involves two components. One is extensive text corpora of neuroscience literature, like BrainGPT using PubMed Central. The other is modality-specific encoders, pre-trained on raw brain signals, EEG, fMRI, megalabrum, for instance, integrated over 2,500 hours of EEG data. Post-training then focuses on the complex task of aligning these measured neural signals with the text embedding space of LLMs. This enables semantic decoding, essentially, interpreting brain activity in terms of language.
Reading thoughts, almost.
Well, decoding semantic content. The tasks and datasets are diverse. Visual decoding uses datasets like things EEG1 and things fMRI to understand brain responses to images. The natural scenes dataset maps boldy signals for complex visual stimuli. For text decoding, collections like Zuko capture EEG and eye tracking during natural reading. Other critical applications include sleep classification, clinical abnormality detection for conditions like epilepsy using datasets like 2EV, decoding motor imagery, emotion recognition. The potential here is immense for understanding and potentially interacting with the human brain at a much deeper level.
That's incredible, using AI to directly interpret brain activity, potentially paving the way for revolutionary neurotechnology. Moving to the cosmos, what's happening in astronomy? It seems like a field with endless data, but also unique challenges.
Astronomy is indeed a fascinating case study, mainly due to its unique data challenges. While it's among the most data-intensive fields, large-scale, openly available, multimodal pre-training datasets are surprisingly rare. Existing resources are often fragmented across text, image, spectral modalities. And data acquisition is costly and slow, relying on long-term surveys like LOMOST and Gaia. Despite this scarcity, existing text datasets like NASA AIDS support models like AstroBird for semantic understanding of astronomical literature.
So they make do with what's available.
They do, and they get creative. In post-training multimodality, models like AstroLava are making strides. They integrate NASA's Astronomy Picture of the Day and Hubble data to generate image caption pairs, teaching AI to describe and interpret cosmic phenomena. Furthermore, synthetic pipelines leverage vast info from ARXIV Wikipedia textbooks to produce millions of domain-specific QA pairs. AstroSage Llama 3.1, for instance, achieved impressive accuracy on the Astromlab 1 benchmark. It demonstrates how domain specialization can significantly improve performance in very specific scientific contexts, even with data limitations.
It's a field where raw data is abundant, but organized multimodal data is scarce, a truly unique challenge. How about Earth science, dealing with dynamic, vast, often unpredictable systems?
Earth science datasets span atmospheric, oceanic, terrestrial, ecological domains. They present unique challenges due to their diversity, scale, and critical spatio-temporal context. Earth SE, for instance, leverages about 100,000 Earth science academic papers, meticulously parsed and cleaned. K2 gathers textual data from internet sources like Wikipedia to build textual understanding. For remote sensing imagery, we mentioned Eagle Vision with eVatcher's 95K, and EarthMarker with the RSVP dataset, 3.65 million multimodal pairs. GeoLove 8K is designed for ultra-high-resolution imagery, enabling reasoning on images up to 8K resolution essential for detailed geological or environmental analysis.
And going beyond static images.
Yes, these models extend to dynamic and multi-source data. EarthDAO was trained on an 11 million pair instruction tuning dataset across RGB, SAR, and multispectral data, allowing richer interpretation of diverse sensor inputs. Crucially, TioChat is highlighted as the first vision language assistant for temporal Earth observation data. It's capable of engaging in dialogues about change detection and time series analysis. This is vital for monitoring climate change, predicting natural disasters, managing environmental resources. These specialized models represent a significant step toward dynamic, comprehensive analysis for our planet.
Wow, the breadth and depth of these domain-specific applications are truly remarkable. But these incredible capabilities don't just appear out of thin air. They rely on massive, carefully curated datasets. Let's delve into the actual development process and the inherent challenges in scientific data, which the paper discusses in detail.
Right. This section critically highlights systemic limitations and opportunities in scientific data development. Starting with data collection and labeling, the paper notes considerable source heterogeneity. Academic and research resources dominate PubMed Central, MIUMP, CXR for life sciences, our GRUs for astronomy, Materials Project, USPTO patents for materials science. These form the primary foundation. These ensure quality and validity, but often introduce significant temporal delays, the data latency problem. Publications lag behind discoveries.
So the models might be trained on slightly outdated knowledge.
Potentially, yes. Moreover, the dominance of English language sources creates a profound linguistic bias. Chinese language datasets, for example, are primarily confined to specific healthcare and agricultural applications, despite substantial global scientific contributions.
That's a critical point about biases embedded in the data itself, which could inadvertently skew scientific understanding. What about the process of annotation? That sounds labor-intensive and needs expert knowledge.
The annotation methodologies in quality control exhibit a tiered hybrid regime to balance scale and precision. Manual expert curation anchors quality in hard domains. Micro-VQA used 12 human annotators for microscopy Q&A. Omni-Earth-Bench used over 40 for earth science. Then you have semi-automated human-in-the-loop pipelines, where LLMs like GPT-4 draft content before human review. Genome Bench did this. It balances efficiency with control. Fully automated systems are used where labels can be programmatically derived with high confidence.
But is the annotated data always representative?
That's the concern. There's a trend toward annotation convenience versus completeness. Datasets often reflect what's easily accessible. Published positive results can be up to 3 times more likely to be published than null results. This bias extends to experimental conditions, often capturing idealized scenarios and neglecting crucial info like experimental failures in material science. If AI only learns from successes, it misses a huge part of the scientific process.
And of course, in many scientific fields, especially dealing with human data, privacy is a huge concern.
Indeed. Privacy and ethical constraints impose additional limitations. While physics and astronomy data are generally open, medical data sets require extensive de-identification, adhering to HIPAA, for example, in strict access controls. This inadvertently creates geographical and demographic biases, as datasets predominantly originate from well-resourced institutions in developed countries. Agricultural datasets face similar challenges with proprietary farming data, limiting the diversity of crops and growing conditions represented. These necessary constraints create gaps.
So these issues can lead to systemic biases within the data, embedding skewed perspectives into the models. Beyond these specific biases, are there broader systematic issues in data quality that hinder Sci-LLM development that the paper addresses?
Yes, the paper points to 3 deeper structural issues beyond specific biases. Firstly, there's a pervasive data traceability crisis. Missing provenance, undocumented pre-processing. It hinders reproducibility and trust. Without knowing the full lineage, verifying reliability is incredibly difficult. Secondly, scientific data latency, which we touched on. The delays in incorporating recent discoveries into model training due to publication lags limit real-time scientific reasoning on the absolute cutting edge. Lastly, and critically, there's a disconnected knowledge hierarchy. Scientific knowledge is hierarchical, but datasets often decouple raw experimental data from its rich context instrument settings, protocols. Crucially, the logical derivation processes and underlying assumptions for formulas are rarely encoded. This limits an agent's ability to perform rigorous symbolic reasoning. And most significantly, the creative aspects of science, failed experiments, serendipitous discoveries, novel hypotheses are almost entirely absent from training data. This deprives agents of the examples needed to learn genuine, innovative thinking. It's a fundamental gap in teaching AI to be truly scientific.
That's a significant gap in how we're currently training these models. So these challenges point to the need for advanced data engineering. What about the technicalities of preparing this data for the models? It's not just about collecting it, right?
Absolutely not. The paper details several formidable technical challenges in optimizing data processing pipelines. One major hurdle is tokenization challenges. General-purpose tokenizers, like BPE, often fail to capture the semantic meaning of specialized scientific terms, complex chemical formulas, long biological sequences. They can break complex names into generic tokens, losing crucial domain fidelity. This necessitates specialized vocabularies and sophisticated tokenization strategies for each discipline. Another crucial step is robust data cleaning and normalization, particularly for unstructured formats like PDFs, which are everywhere in scientific literature. Figures, tables, text must be accurately extracted and converted to a uniform input format for efficient model processing. This often involves complex OCR and layout analysis.
And LLMs are inherently text-centric, but much of scientific data, as we've discussed, isn't text. How do we represent that non-sequential and non-textual data so LLMs can understand it?
This presents a profound challenge. Representing non-sequential and non-textual data for text-centric LLMs requires innovative approaches. In chemistry, models must interpret 3D molecular structures inherently graphical alongside text-based smile strings. Similarly, protein structures, gene regulatory networks, genomic data in biology. Challenging to represent in a standard linear transformer. Addressing this requires multimodal or hybrid architectures. Some approaches use graph embeddings for structural info, then project these into the transformer's input space. Other methods rely on specialized encoding schemas, representing complex equations or tables as structured text sequences, carefully preserving logical relationships. The challenge lies in ensuring these representations maintain semantic fidelity and allow the model to truly reason across modalities, moving beyond simple text understanding to grasp complex scientific relationships that aren't inherently linear or textual.
This isn't just about automating existing tasks. It seems like it's about pushing the boundaries of discovery itself. So what are the ultimate objectives and the critical considerations outlined in the paper for this brave new world of scientific AI?
The paper concludes with two critical areas. First, the objective of from SciLLMs to scientific discovery. The ultimate goal extends beyond automating routine tasks to actually accelerating pivotal scientific breakthroughs. SciLLMs have this unique potential to identify subtle, non-obvious correlations and patterns in vast multimodal datasets that would be impossible for human researchers to process quickly. We are transitioning to a stage where these models can serve as powerful instruments for accelerated hypothesis generation, potentially contributing to, Nobel Prize-worthy discoveries. While human creativity and ethical oversight remain vital, SciLLMs will act as collaborators to significantly reduce discovery cycles, potentially redefining the scientific method itself. It's about supercharging discovery.
Supercharging discovery.
And the ethical side. Secondly, yes, the paper stresses the need for ethical governance for responsible scientific AI innovation. As these systems influence research directions and resource allocation, ensuring equitable access is critical. We need to prevent the concentration of scientific capabilities in only a few hands. Questions of attribution, who gets credit? Accountability, who's responsible for AI outputs? Validation for AI-generated discoveries. These require careful consideration and community consensus. Furthermore, the environmental impact of training these huge models must be balanced against their potential contributions to sustainability science. This demands innovations in efficient training and architectures. These are not just technical considerations, but profound societal ones needing ongoing dialogue. #tag outro.
Wow, what a deep dive into the fascinating, complex world of scientific LLMs and MLLMs. We've covered everything from the fundamental data modalities underpinning these systems to their cutting-edge agentic capabilities, the rigorous evaluation frameworks needed, and the profound challenges ahead in data development and ethical governance.
The paper clearly illustrates that SciLLMs and MLLMs represent a fundamental paradigm shift in scientific research. They're transforming how we interact with data and conduct discovery. We've seen how advancements in data, model architecture, coupled with stringent quality standards and specialized evaluation metrics, are enabling AI to tackle increasingly complex scientific problems across incredibly diverse domains: chemistry, biology, astronomy, and more. The progression from general LLMs to specialized AI co-scientists really underscores the incredible potential. It also highlights the critical need for addressing biases and ensuring interpretability and responsible governance.
It's abundantly clear that the future of science will be deeply intertwined with these powerful AI tools. This paper truly gives us an invaluable roadmap for understanding this complex, rapidly evolving landscape.
And considering the rapid evolution detailed in this paper from basic text understanding to autonomous scientific discovery, we're left with a profound question for you, our listener, to ponder. How will the increasing agency and self-evolution of these scientific AI systems fundamentally reshape the very definition of human scientific expertise and creativity in the next decade? Will the aha moment still be uniquely human, or will AI agents increasingly share in those breakthroughs?
A truly thought-provoking question for you to mull over. Thank you for joining us on this deep dive into scientific LLMs and MLLMs. We encourage you to continue exploring these critical developments and the fascinating future they promise.